Okay, so we will continue to practice exploratory data analysis using MATLAB and apply this to single-cell RNA-seq data sets. So as I mentioned from last week, and you should have this installed. Okay, if you haven't yet, you need to visit this documentation page. The keyword you want to search in the Google is scGEAToolbox. Toolbox is MATLAB package. They like to call it toolbox. It's a collection of functions developed in MATLAB language for a specific task. In this case, it's for analysis of a single-cell gene expression analysis. Okay, so that's why it's called scGEAToolbox. So this toolbox has been published. If you want to know the paper, and that's the link you can click, you find the paper published 2019 in bioinformatics to describe the basic functionality of this toolbox. And since then, there are a couple of other papers or authors have been using this. So there are citations for papers citing this paper, okay. So I keep this updated recently. So for education purpose and for end users have a quick reference. For example, if you haven't done this, and just copy the code here, and this will go to the web page. This web page, download the package, unzip it, and also add the parse. After that, if you search one of the functions inside the toolbox, you will be able to find it. Once this file is found, that indicates the toolbox has been installed successfully. So just make sure you have this. And then if you are ready, and we can just start to use MATLAB environment to do this.
Yeah, I think we installed that toolbox last week, so I think we'll be ready to go.
That's good. So we are on the same page. That's great. So try this first, CDGEA. So CD, this is a function, and I just give a name like this, which is a change directory to gene expression analysis tool. So if you do this, you should be on this, in this folder. If you are not in this folder, it will be switched to here. Okay, there are several places you can start. In the documentation, I have this section, see, getting started. So this getting started will help you to bring up this application called the scatter plot to load example data I already prepared, and then quickly you will have this. I think last week we showed this, but we just go through that step. Okay, Bowen is here, and Bowen, and if you can just bring up your MATLAB, and you should have that last week installed this already. We are ready to go.
Okay.
And if you under this folder, you see this daemon underscore getting started. That's what I try to let you to run it. In order to run this, you can just type that command in the file name underscore getting. MATLAB is case sensitive, so you want to keep this uppercase and lowercase. And oftentimes, if you use the type key, you should be able to automatically complete this. Okay, so you just press return and the show up the welcome page, and the first step is to use this to change the current directory to where we are here right now. And then you can press any other keys to continue this section. So the second step, you don't have to type anything because this automatically runs this code, but I show you what it's doing. So in this second step, we are loading. What are the line of the code? What was the line of code? Yeah, so you just use this to bring up because there's a function already here. So this is just like a quick start. You don't even have to type this in the command line. You already let this script to run. So we already have the first step, second step. Second step is to load this example data in. So we can click next button. Okay, you load that, and then you use this scatter plot because you load the expression matrix, the gene list, and also coordinates of this embedding. I hope you still have some impression from last week what they are. And the purpose of this demonstration is to let you to quick access to this interface, okay, without any program. So you know you can do this. I think that's exactly the data last week you have already seen. So the last step in this demonstration is to load not only this, but another object. So this object is a composed variable, which include all of this information and also something extra. That's why you can give the color and the separation of clusters. Okay, and then I think that's the last step. This show you another function which inside the tools. Okay, I don't know if you know the embedding. We introduced the concept of embedding. And you can use a t-SNE, you can use a PCA or UMAP to lower the dimension. So you can have a three-dimensional plot to show the relationship between cells. And if you apply two different embedding algorithms, such like a t-SNE on the UMAP to the same data set, you end up with a different embedding results. And this tool will help you to look at this embedding side by side, which produced by two algorithms. There are same group of cells, but because of the embedding algorithm it applied differently, so you have a different distribution of these clusters. What this tool will allow you to, for example, to select this group, you're supposed to have a corresponding cell which have the same from the same source, or exactly the same data, and it be highlighted here. So this tool allow you to compare the two embedding results. For example, if I want to know what cell in this group are in this embedding results. So you can see you map group those cells into this isolated cluster, but it's not t-SNE. t-SNE still put them close to this cluster, but also separated some of those cells. So for the same data set, if you use different embedding algorithm, you got a different results, but these two allow you to compare. All right, so this cluster is this cluster. So these two embedding algorithm basically report the similar results. You have a two larger cluster cells. One is this or that, another should be this group. Okay. Okay, so this demonstration just tell you how to do it and which function you use to do this. For example, this embedding comparison is a function called GUI.SC multi embeddings. Okay. For example, you can copy this command line. And if we close all the windows, we just opened and clean my desktop, you can still use this command. You see, we have no windows, but you still can use this to visualize this example. Okay. And if you are interested in what's going on here, you can do the edit and this. And you will see this is a function developed inside here. So you can provide two embedding coordinates and the title for the method. So if you don't provide anything, so the input argument is smaller than 0. So basically you didn't put anything. This will be automatically load an example file to show you. That's what we are having here, to load an example file. Okay, so this is the getting started. That's the source code for this application. Are you okay? So we are done with this small demonstration. So that's what you see on this page. So you may have a question. If we have a example data, for example, X, you know, is gene expression level of the matrix, and then we have a list of gene. How can we make this SCE object? So we can use SCE object in the tool instead of individually you put a lot of variables. That's the function that you can use to combine these two together. Combine these two together into a new variable called a single cell experiment. So basically that's a data type hold the most essential information about your single cell data. Okay, so to make this class, it's called class or object, you need to have X and G, okay, the gene list. By the way, if you like to try, you can copy this. Okay, so that's change the directory, load the example data. You have X and G, that's all you need. Then you make this into a SCE object and then put SCE in the scatter plot. You will get something like that. So give it a try. I think we should do it.
You said that the CD stands for change directory.
Yeah.
Does that like put like a new data set up front?
No, see, this just make sure you are in this folder, because this folder, you need to go into the example data to get your example. So this is relative parse. So I just want to make sure you are in the folder, this toolbox is installed.
Okay, thank you.
Yeah. I think that's about the application of this scatter plot. I try to sell this to you because it's very easy to use. You have a nice interface. You can do a lot of exploratory analysis on it. So from since last week to this week, I'm trying to show you this. Okay. So I will give you 3 or 5 minutes to try this out. Make sure you understand each step. If you don't, let me know why this scatter plot need 2 or 3 variables. What the variable mean here? Can anyone explain this to me so I know you understand?
Does it need the 3 variables for each of the 3 axes, so the X, Y, and Z for that 3D feature?
Yes, so the X, Y, Z coordinates is in the S. If you look at your S variable, it should have 3 columns. So you look at it on my screen. If I open the S, this S has 3 columns. That's X, Y, Z position in the 3D plot for each cell. Okay. So the X has a dimension of these numbers of a gene, 2,600 cell. So that's S have 2,643 rows. Each row is a cell position. And that's a name, gene name. Okay, for your project, I hope you can find a way to get X and G, right? Once you have X, which is expression matrix, and then you have a list of gene, your project will be almost finished, because once you have these two variables in there, you can bring up an interface like this and do all the analysis you want to do. Unless you really want to know what's going on exactly, you can open each of these function and look inside. Okay, so if you want to know where I can get X expression matrix, where I can get list of gene, you may want to look at this code formulas. Okay, because in the getting started, I use example data. You just load what I already prepared for you. But for your own project, your task will be go out to find interesting paper, to locate where the data, published data is located, and to find it online and find a way to download it. So that's where I put example code here. For instance, the first section about this code formulas is to import data from a download geo dataset. Okay, I shouldn't do, let me see. Maybe we should jump to these tutorials. So I think I will reorganize this tutorial and the code formula. Let's jump into the case study and the tutorials. So you will see here, the first item is download the data from the Geo. Geo is a database. And it's NCBI database. And oftentimes when authors want to publish their results, they need to upload the raw data into the Geo database. So they can register for their dataset ID, something like this. So this is a sample ID. So allow other people to use this ID as a key to identify, to locate their data and for download. For this example, I gave this particular example, which published by a group of authors. If you go into this link, and you will find who published this, and they report the result in which journal and which article. And if you browse down this page, you will find a list of information of the files associated with this sample. And if you read the description of this sample, it has been subject to single-cell RNA-seq experiment. So this file, those files are created from the single-cell RNA-seq study. If you look carefully, there are 3 files. The first one is so-called barcode. And by the way, this is sample ID, and the name, the author gave a name of their sample. So this sample is it's kind of endothelium cell, which inside the lymph vessel. I don't know exactly which part of this endothelium cell, but it's in the lymph nodes. They extract those. And this first file has the barcodes, which is the 10x genomics barcode for each individual cell. And then they have a list of genes. And then they have the matrix. Okay, so the matrix and the gene are the essential information we will need for prepare our X and G in order to load into the toolbox. So you have a visualization, you have a analysis. Okay, once we locate those files, we can right click and copy the link to the file. We want to use FTP download. And then that's the URL we copied from those links. And then in MATLAB, you will need to use this GUnzip because all these files has been zipped. GUnzip, you will unzip them automatically and download and unzip them. So that's what you need to do. You locate the file, copy the URL for their FTP download, and put the URL for as an input for this GUnzip function. And then if you run those in MATLAB, you will download and unzip those files. So if you do this successfully, you should have 3 files in your folder you are working on. The first file will be the matrix.mtx. The second file, let me see. Okay. Okay, depends on the. So I don't know after you unzip what's the name of the unzipped file we will call. The unzipped file maybe this. So I'm going to give it a try. Okay, you can try the same as well because this is a good example you can try because I want to know after unzip what the file name is for each of these. So I will rename this because this function will take the output of this. You need to provide the correct file name. If everything goes right, you will have 3 outputs, X and the gene list and the barcode list. All right. And in our analysis, barcode list is not essential. It's just a barcode for each cell. In some other so-called multimodal analysis, you may need this to match the cell from different experiments. If you have a cell which undergone several different batch, but in our case, it's not essential. So in just this one step, you use this gradient function, and you should be able to get your X and the gene list. And then you can put that in and you will see them. So I'm going to copy this and paste in my MATLAB environment and see what file I will get from this download, okay? I'm going to use another less important folder to save my file. Feel free to copy this and to download in your own computer. It will take some time, I believe. Especially the last one. This is a huge matrix. On my computer, they still show busy. I see 3 files already. Well, I renamed it wrongly. So anyway, I have 3 files here. So I have to copy this and replace this name. So the barcode is not the barcode. Should be the correct name should be this. So I just download the gene list. This. You see, I'm replacing the wrong file names. To the one it's supposed to be. Okay. I don't know if you have this already. I'm going to look at the gene. In the genes, I right click and open as a text. I see the 2 columns of genes. The first column is ensemble ID. And the second column is a gene name that makes more sense to me. So I'm going to use the second column as a gene name. That's why in the following this function, there's 2 here that indicate I want the second column as my real name of gene. If you don't give this 2 here, you can leave it blank or just ignore it. Then you will get a list of genes look like this. It will still work, but people tend to look at the real name of the gene to have a quick impression of what the function of those genes are. Any questions? I see Mark join in us.
Yeah, sorry about that. I was taking an exam on Galaxy, and in the middle of it, someone had 1,000 jobs go through, so that delayed the entire exam.
Okay, no problem, no problem. Welcome. Okay, so I'm updating my documentation here because I found this mistake. It's a mistake because it's not a fit with what we this example. Because after undep, you have 3 files. You have to name them correctly in order to allow this function to find it. It will take some time to allow the system to refresh. Still not yet. What I'm doing here, we just wait until my documentation become correct. Download, still not yet. Okay, so after a couple minutes, so you see, I just changed my documentation here. And now the file I'm going to load is exactly what I'm downloading. I just downloaded and also unzipped. So this code should run. So I will copy this. So if you don't have this, you have to wait and refresh your web page because I just changed this on fly. I copy this code and go back to my MATLAB because I already have these 3 files in there. So I can just paste this. So this is a really long name, so it takes space. So that's why I put 3 dots here. So allow me to change to the new row. Okay, I'm running this in order to read information from these 3 files and put into 3 variables. That's our input. By the way, you can click in the white space here to open the current folder in Explorer. So I want to open this because I want to see the size of this matrix file. Can you see on your screen? This is a 185 megabytes. I can use one of my text editor to open it, and you will see what's in the raw data of this matrix file. So this file is named matrix market format. Okay, so that's what you will see. The values of expression and the coordinates. So this file will be translated into X and the gene list. Now I'm done. My X is here. So this is a huge matrix. Can anyone read this, the dimension of this? More than 32,000. And that's the name of the genes, the number of genes. And we have 8,651 genes. Okay, that's the matrix we are dealing with. And your gene list is this one, 32,000. Okay, so this column come from this file. The gene list is 1-to-1, okay, match. So this first one is mRNA gene, microRNA gene. So you have this here. The second is this, open reading frame. All right. Do you still remember what we did last week once we have this big X matrix? Remember we did a sum of all the rows or columns in order to see the total rates have been sampled for each gene, for each cell, so cells by column. And we also can look at the distribution of this so-called library size. So this allows you to see the library size, because once you sum them, you will get a list of 8,651 values. So let me do this 2 steps. So this is called the library size. Okay. So library size is this. It's the sum of the column. So the first cell, for example, fourth cell is first column. When you sum them, you get 2,827. That means there are 2,827 rates has been mapped in this cell in different gene. And the sum of the cells you see looks like luck here, okay, like number 16. It has almost 3 times rates sampled in cell number 16 compared to cell number 1. So this creates some kind of unfairness. So we trade technically, because of random effects of the sampling, we most successful in regarding the number of rates measured in some of the cells, but not the others. So we want to look at how severe this problem could be. So we can do the histogram of the library size. We just have a list of this, the library size. If you do a histogram, you see something like this. So the average could be 5,000 somewhere or less. And this is 10,000. And this is 20,000. So the highest value may be more than 60,000. So one of the cell has been maybe sequenced very, very deep just by chance. Or maybe the cell is larger than average. But that's a distribution, give you some idea. Okay, my data, the smallest is between the bin age 1,500 to 2,000. Some cells sequence is shallow, and some cells are higher. And you may want to use some cutoff. See, if it's too high, I will get rid of them. If it's too low, I want to get rid of them. So you have a selection. You have a way to trim off and discard some of these suspicious, highly sequenced cells. Okay, all this has been considered in a quantity filter. So a lot of this can be done, just uses filter function. This filter function, if you look, it takes into 2 variables, the big expression matrix and the gene list, and throughout the same gene list and the matrix is only just want to save some the memory space. You can make this X2 or gene list 2. You keep original. But in this case, I just replace original and with a smaller, cleaner data. So after this filter, QC filter. So I'm going to do this. Look at this dimension. We have a 32,000 cells, genes, and 8,000 cells. So after I finish this step, the dimension this will be smaller, shrinked. So let's see how many genes or how many cells have been filtered out. filter them away and discard. So I'm running this. Now I have a, after the filtering, I still have 11,600 genes, and still have 8,581 80 cells. And correspondingly, the gene list also becomes 11,600. Okay, so I finished the QC. Now I'm going to select some genes. So this is called select gene based on some of the cutoff, because I still have too many genes to study. So this function will also take expression matrix X and the gene list, and I set 2 parameters here. So this one means I need to find a cell to be expressed at least once of that gene. And then this is 5% of a cell has to be detect at least one expression of that gene. I will keep those genes. So you can imagine if I do this filter, some of the gene express only 4% of the cells will be discarded because it's selection of the slightly highly expressed gene. So now you have 11,600 genes. So I'm going to apply this step, the filtering, to select some genes. Now I only end up with 7,288 genes. And this function will not touch on the cells, so still have the same number of cells, only get rid of a lot of genes. And the gene list becomes smaller as well. So those are the gene unselected, has been filtered and selected. They are relatively high expressed among the cells. You have to be expressed in 5% or more cells in order to have to be kept. So now I'm going to open this. You start to see the ones. Start to see the expression values, because this has been filtered by the relative expression level. Some of the gene detect everywhere. Okay. Okay, so once you have the gene list, you have a big X. So we will go back to what we started with, because once we have the list and also the matrix, we can start to use scatter plot to work on that. And there's no embedding because you have to recompute that. There's a function which is here to allow you to recompute embedding of cells. You can select one of the approach. Okay. So I will leave it here and we come back in 5 parts 3 to continue. Okay. So I'm going to click this to allow the algorithm to run in the backend, and we will come back. All right, any questions before we take a break? Okay, I don't know if you have tried this out. On my computer, I have a really relatively new desktop. It took me, so that's about 6 minutes to finish this UMAP embedding for a dimension of data with 8,580 genes, 80 cells. Each cell has more than 7,000 genes expression. And I get this very funny shape distribution of cells. Any one of you tried this?
Mine is still running.
Okay, are you using UMAP or something else? You have 3 options.
UMAP.
UMAP, okay. Okay, so let's see what we can do if we get something like this. Of course, if I download this data set, I need to read about the experiment procedure and see whether this is a human sample or animal sample, which tissue, which cell type. Of course, I remember this epithelium cell in the lymph nodes. Let's see, I will suspect majority of this cluster will be T cell. No, not T cell, endothelium cell. So maybe some T cell in here. I don't know. So I'm going to give it a try, use the function inside the scatter plot. I will start from this purple group. I highlight this. And this small brush here tell me I'm going to search a database, the built-in database, a small scale database, and see what's the cell type of this group. So I'm going to just click this button. So label the cell type of brushed cell. I say yes. And ask me about the species. Do you have any idea this sample is from a human or from a mouse? We can go back to check. My memory tell me it's a human. So I'm going to use this database called BangaloreDB. And the program just search the database, try to match the marker genes and tell me the potential gene type, cell type. So on the list, the top one, endothelium cell, the second is the fibroblast. The third one, just some names, Bergman glial. So definitely not the Bergman glial, not the astrocytes. So the first guess, we have this endothelium cell, could be the correct one. So I just select this, okay. So once you select this, the cell types have been marked and put a label on it. Then I'm going to see, let me try this group. It's orange. What is the cell type of this cluster? So just do the same thing and click yes, see human BangaloreDB search. Okay. tells me this hepatic stellate cells. Can anyone Google this cell type and see whether it makes sense for our sample? So I'm going to search so what they are. Okay, so I don't think our cell is from the anything to do with the liver. So I will reject this option. Maybe it's a fibroblast, maybe it's another type of endothelium cell. I don't know. So, but for now, I just mark it as a fibroblast. Okay, it's close to the more general options. Then I'm going to move into this huge cluster. I will start from this tail and see what's special about this. Just select this and press human BanglaDB. Well, give me some hint. That's T cell. Endothelium cell. Now we are running into some most difficult stage of the study. We don't know the samples very well, and the database suggests using the marker genes a very diverse set of potential cell types. So we don't know which one to select. So let me just stick with the top selection and see this T cell. I'm going to try another group here. This endothelium, this part. Endothelium. Okay, so I will feel comfortable about this, the quality of this data, because it's a match with what we expect to see. Because if you look at the record of this experiment, they collect these endothelium cells in a particular way. They have a FACS selection. They sort those cells using FACS. Flow cytometry. So there was blood cells in depleted. So we didn't see many blood cells in there. Live cells are known. Sorted. So media count. So from reading the study of this, we are okay with the majority of this cluster of endothelium cell. And this fit our expectation very well. And so far, I think we did a very good job to clean the data, download, and then visualize them relationship and confirm the majority of them are endothelium cells. Okay, so those special this tail, we don't know, maybe some contamination from T cell related. All right, are you happy with this progress so far? Any questions?
No questions from me.
Okay. Any, have you got this similar UMAP or you use T-SNE?
Yeah, it looks about the same.
Same, oh, okay. Okay, we go back to this tutorial. And these are the standard protocol we can use to process 10x genomics raw data. So the raw data, what I mean is 3 files. One is matrix file. Another is called features or gene list, basically. And the barcode. That's the procedure. You can just repeat this for many different data sets. Just make sure the name of the matrix file is correct to match what you downloaded. And you know what it means by two means second column of the features as the name of gene. And then this scene and the standard way to filter and also enrich those highly expressed gene. And here I did a normalization, use one of the method to normalize the library size. Okay, remember some cells have been sequenced many times more than other cells. And this function will identify the highly variable genes. Those genes are more informative to tell the different clusters sometimes. And the selection of the top 2,000 highly variable gene will make this embedding faster because you use more informative genes and get rid of a lot of less informative genes. And so you only take 5 minutes to do it. Maybe this will only take 2 minutes to finish. Okay, so this is a pretty standard way to process those files if you have 3 files download. The next section of this documentation is overview about this tool. So you have we have tried this brush, for example. There are a group of the icons here share the similar function, which about the cell type. You assign cell types to brush the cell, or if you have a cluster, and this button will assign the cell type for each cluster of cells. And afterwards, you can rename your cell type. You can find the marker genes. See, the cell type for this cluster, which genes will support that. You can have another option. So this cell type related function. Those are the clustering related labels. and there are pseudo time related functions here. The differential expression, okay, you found the genes differentially expressed across cell types or across clusters. There are network related, you build a single cell gene regulatory network. So this is a newly added function. So I'm still working on some of this function to make this stronger. Okay. And most recently, I put another section, it's called scGEAT R package. If you have a result from there, then you can transform the scGEAT package results into this matrix file, into the feature file, into the barcode file. Once you have these three files, you can load them again. Use exactly the same way. So in this way, you output the R object, scGEAT object in R into the files, and then read the file into MATLAB. So this gave you a way to import scGEAT file into MATLAB. Okay, so the example file I use is download from here. There are two scGEAT R data files, and you can use this way to convert and make use of MATLAB functions to further explore it. So that's the section already finished, so you can take a look at those. Okay, it's time for question.
Do we have to rename the files every time?
Yeah, so it depends on you download the file and you unzip them and what the file name is. If the file name is this, you have to replace the gene list file which match the file download, unless you want to rename the file after it download. You rename to feature, rename it to matrix, rename it to barcodes, and then you can just copy this and we'll do the job.
Okay, thank you.
Yeah, otherwise you have to name them correctly. So for your project, you should go out, go to the geo database, to search the keyword and for the cell types or samples you are interested in. Or you have to go to the literature to find the paper, and the paper then you search for the geo accession number. So for example, if I go I use single cell RNA seq in my keywords. If I want to do give me a system, then maybe astrocytes. Okay, I'm searching for the paper using single cell RNA-seq to do astrocytes study. Okay, for example, I found one paper published last year come into this page. It's not a very good journal anyway. Then here I'm going to search the geo. I hope they have this. Okay, so this paper, they may not release their data, and I don't know where the data they put. So I read the paper and see whether they dispose data somewhere. Unsuccessfully. Another paper is published too early, so I don't want to use it. Sale, and then okay, this is relatively results human map, so 3. So I just see whether this will give me some access number. So I will search geo again. Okay, I found this ID. And so this is called a single nuclear. Okay, it's similar to single cell, just slightly different preparation because the astrocytes or central nervous system neurons is too sensitive to the damage of the cell preparation. So for the safe, they just extract nuclears from the cells to sequence them. It looks like this will the place they put their data in, so I can use this accession number to locate this data set. This is one, 299. Well, they put a lot of cells in here. So this is not ideal. It's not a standard. Looks like they put every single cell data in there. They're separated. So this is not a well-prepared samples. So I will not go into that. So I'm going to switch to another study. Keep looking. Okay, something like this. This geo, this paper. Yeah, I really have to read description, and so we can have idea of what they are. So I tried, you see, I tried a couple of this search, but not successfully identified any good, well-prepared data set for use directly. So I will keep looking, read the papers. So this neuron stem cell may be related. So I come into this paper and then search for geo, nothing. Yeah, probably they didn't do any single cell study. Yeah, so we have some session numbers, but we have to look into each record more carefully before we download anything. Okay. Okay, in this part of the annotation, the documentation, we have a more structured code formula that you can use. For example, import files downloaded from Geo. And if you download this, you have the correct name of files, then you load them. Okay. If you don't have the barcode, you don't need it. If you only have two files, the expression matrix and the gene list, so just keep this as an empty, and then you should be able ready to go because only the information about expression and the gene list is used. And this is a standard way to process filtering and inviting. Extract top 2000 highly variable genes. And this is a relatively complete pipeline for raw data processing. There are normalization, extract feature genes, and embedding, and then create a CE object, and also estimation of the potency, estimation of a cell cycle, clustering, and then scatter. Okay, I don't know if you have any questions. So I'm going to clean up my desktop. So I clear all. So this will get rid of all the variables already in the workspace. Shouldn't be so slow, and I don't know why my computer is frozen. So I just bring up a new fresh MATLAB environment here. And still I'm going to go into my MATLAB toolbox folder. Here I have several demonstration scripts. So those are the command lines. So if you don't have the interface, you should be able to do it using command line. So I will double click this file and open it. So these files have been prepared and also annotated, so you can see the section is separated. If I highlight this section and the background of this within this subsection is highlighted. If I click another section and then another section is highlighted. Once I highlight it, I can use the run section instead of run the whole scripts. I run sections one by one, so I can demonstrate the steps of the analysis. So I'm going to go ahead, the first script file, including select of genes. So if you have two data sets, you want to find the intersection of the genes of the two data sets and the visualization. So I'm going to click each section one by one to show you how to use command line to do some jobs. This gave you the flexibility. You have some function you just cannot achieve using the interface, and you can do it in this way. So I click run this section. I highlight the first section. Okay. So this section will just change the working folder to current folder, and then use this function to read two files. So these files are prepared in the CSV format, and you can open them using Excel. And here, those information is read in, and for each of them, you have the gene expression matrix and the gene list. One is named big X, another named Y. Okay, so you can separate this. And this two data sets from a single cell RNA-seq, but they contain different number of genes, so they're not match. So I will, in this way, I will first select the genes by some of this cutoff. At least 3 cells have more than 5 reads per cell. So I do this independently for X and Y. Okay, after select the gene, you still have over 6,000 and over 5,000 gene included in 2 samples. Now here I use intersection of another built-in function to find the gene list shared between gene list X and gene list Y. And afterwards, I will select those genes so X and Y match to each other. So, okay, I prepare the XY, and they have a shared gene over 5,000, and then they share the same list. So the rows are the same gene, okay? So I can combine them, concatenate them. And in this case, I want to show the raw data. So this image will show the raw data points. I certainly can look at the matrix. I open and look at the matrix, something like this. And the Y is like this. And for this one, I show an image. It's a heat map. Okay, like this. I select 100 genes and 500 cells. So this is X and that's Y. And this is raw data, not scaled. So gene number 3 have a relatively high expression level across all the samples. So you see the light color, that's gene number 3. If you look at the variables, number 3 expression level really high. So you see the light line here. And the rest of the cells or genes expression level is relatively low, this several hundred times low. So that's why you see a lot of this blue. And one of the gene, maybe number 81, have a high expression. Okay, so this gave you the overall impression of your raw data. So I can see what's the number 3 gene, what's the number 81 gene by looking at the gene list. So number 3 is this gene, IGS 15. Number 81 is this gene, ribosome protein, large unit, number 11. We have a special gene, okay. Okay, so I can keep going, run down to the normalization. So I apply normalized function, normalization function to these two data set independently. Then I will watch that image again. So this is a normalized values. Okay, after normalization. And then there's another way to normalize the data. So that's one way to use D-seq normalization method. And this is using standard library size normalization. And then I run that again. So that's our normalized data looks like. And do some transformation. If I do a log transformation and do a expression level +1, the reason is a lot of zeros in there. And after the log transformation, you start to see the magnitude difference between the highest expressed gene and the lowest expressed genes are smaller because after the log transformation. And still you have number 3 genes, the highest expression level is yellow, and number 81 is the second place. And you start to see this pattern. And then there's a imputation method called magic. Because we have a lot of zeros here. You see these zeros, even after the normalization, we still have zeros in there. Imputation method will take care of those and try to fill in that values, use neighbor values. So I can run this very quickly. You got imputed results. And the outcome is in the XO. So you see all the values have been filled in. So that's an imputation. And another way to do the imputation, and then there are batch effects correction. So if you have a X and Y, you want to merge them and make them more close to each other, you can use this. yeah, basically that's all the functions in demonstration number 1 is all deal with this raw data and do some basic transformation. If you need imputation, you can do imputation. Okay. So this just gave you the impression of how to use command line to do this job. I will stop here today, and you have several scripts. So now we only touch on based on the number 1. You have 2, 3 to 6 for the demonstration, and you can play with them. If you have any problem, if you found any bugs, let me know. Maybe someplace the code is not correct, and you see the breakup of the progress. Just give me a message. I will fix it. Okay, I have to close this session in 5 minutes. So if you have any quick question, please let me know. Otherwise, I will see you next week.
Okay, thank you.
Okay.
Thank you.
Yeah, so the lecture is recorded, so I will send you the link for any the recording for the reference. Okay. Good.